Hi class, and welcome to chapter 3. This is the first lecture. I expect there to be about 3 on this chapter. So this means we are starting week 2, and you have completed week 1 for lecture and lab. Guess I'll see that soon enough. I hope you learned something. I hope it was okay. There's still time to add drop. If it's not okay, you have options, but only through this week, I believe. We'll see. So hopefully you've met me in person because you were able to come to lab, or you've already met me in a past semester. I'm really looking forward to having the face-to-face time, so I'm happy for that. I hope it lasts. We're going to keep our fingers crossed. But continue on this path. So, having said that, our next chapter, this one, is the beginning of chapter 3. And chapter 3 is about the cell, and we are not doing all of that chapter. It's a whole lot of just ton of stuff. And I'm peeking at some of my notes. We're doing the first 3 sections. I'm not sure if it's the first 4 or the first 3, but my notes say 3.1 to 3.3. And after today's lecture is going to be about just an overview of the cell and talk about the nitty-gritty of the plasma membrane. And then we're going to be talking about transport in the cell. Those are the main points of this chapter that we're focusing on because that's the basis of how our body works. So, having said that, let's get going. So, all right. So, yep. I'm trying to see how I can put this into slide presentation. Okay, looks like everything's all right, and I'm still in the active window. Oops, I don't know, I'm going to just put myself, I don't know, I guess I'll put myself right there. 3.1 to 3.3, so just a part. I mean, this whole chapter talks about the cell, all the organelles, mitosis, meiosis. We're not going to do all that, so yay, that's too much. Having said that, when we're done this week, you have an exam next week on chapter 1 and chapter 3, so because we only do a little part, no problem in getting an exam on it. Okay, so having started, the cell is just crazy to me. The cell is the basic unit of life, right? And if you think about that, okay, yeah, we have millions of cells in our body, hundreds of different types of cells, there's cell specialization, but let's just think for a minute that there are things smaller than a cell that are greatly affecting our population. And I'm talking about viruses. So viruses are not even considered a living thing. And, HIV virus has affected the growth curve of the world, and now we're in the COVID-19 virus, which is greatly impacting our lives today. So how crazy is that? That's not even alive, and it's able to make such an impact. That messes with me. All right. So over 200 different types. So let's look at some of the cells of the body. And I'm going to expand this a touch because I see it's not in. Can I grab it? Can I grab it? Gonna let me? It's not going to let me. All right. I'm still learning this program a little bit, and I see once you start recording, you can't really do too much. All right. So if we look at this, we have red blood cells. They're erythrocytes, and they are biconcave, little flat discs shaped to have awesome surface area. We have boring epithelial cells. I'm not looking forward to the next chapter. You have skeletal muscle. They're long and thin. Like when you eat meat is muscle, and if you're like, oh, this is stringy, those are different muscle cells. White blood cells that have crazy shapes. Fat cells. We have a neuron here, a nerve cell, and even a little single cell, half a cell really, of a sperm. So all different shapes and sizes, and that contributes to how they work. You've evolved to have this, and they all have a very specific function, so they look like it. All right. So when I look at a cell, this is like the cell, right? You've got the outer plasma membrane, and that's the part we're really going to focus on. Inside, you have the nucleus, and the nucleus is full of DNA, chromatin, chromosomes if they're replicating, chromatin otherwise just kind of loose, and that's your genetic information. An endoplasmic reticulum with ribosomes would be rough. Smooth endoplasmic reticulum would be somewhere else. You have a Golgi apparatus, the UPS for packaging of the body. Mitochondria. See, I'd be asking you all these questions if we were face to face. Mitochondria is the powerhouse of the cell, right? That's what everybody knows it as. What does that even mean? That's where you get the biggest bang for your buck in making ATP from oxygen, aerobic respiration. What else do we have? Little lysosomes we talk about. Centrioles assist with cell division. You could have projections coming off like a little cilia or a longer whip-like tail like a flagella. Sorry, I gotta get a drink here. And different parts. So we're going to talk about different parts of the cell as we need to. There's certain tissues, certain systems where we will talk a little bit about some specific parts inside a cell, but that's not this chapter. This chapter is really about the plasma membrane and transport. That's what I care about. All right, so when we talk about the cell, it's the plasma membrane. Kind of important. The nucleus that has your chromatin, which is your genes, and then everything else in the liquid part of the cell, I guess you could say, the cytoplasm that has your organelles. All right, well, that's 3.1. So when we look at the plasma membrane, I'm going to have to get my little pen here. I just want to see something for a second. Okay, does it talk about that? Okay, it does talk about that. I didn't look at this ahead of time. But we didn't do chapter 2, so there's a little chemistry that I have to talk about in this, so I just wanted to peek ahead and see, okay, what chemistry do I have to talk about? Hey, come back. All right, so this plasma membrane is a beautiful thing. It's called a fluid mosaic and has all these different components. What do I want to say? So there is, and we're going to go into details, I guess, in the next few slides, but let's just, can I zoom in on this? Oh, I can. I'm going to get pretty fancy here. Now I need to move it up. Okay. So like I said, remember this whole, but then I can't write because I've changed the size. All right, we're back to where we started. So we're talking just about this part here and all this stuff that's contained in it. And this part is just this outer barrier of the cell, right? That's the plasma membrane. So the plasma membrane is a barrier and it separates the inside, this is the inside the cell, from the outside. And the main thing I'm looking at are all these little blue guys with two tails. These are called phospholipids. Oh, it says it right here. And they are these guys. Oh, I don't mean that guy right there. And it's a bilayer because if you see, bilayer means two layers, right? They're oriented with heads. So this part here is, oh, you stop. This part here is a head, and then it has like two tails. So it's like a phospholipid head, and then these two little tails coming off on it. And you see tails are oriented towards each other. Heads oriented to the outside. Tails inside. Tails in the middle. We're going to talk about why that's a big deal when we look at that one slide that I just peeked to. Okay, so you've got these phospholipids, and then you have proteins. You have proteins that cross outside to inside. Those are integral proteins. So one end would be on the inside, the other end is on outside. There's a whole lot of different types. We're going to talk about that in a later slide. And you have proteins that are just either on one side, either on the outside, or they might even just be on the inside, like this crazy guy here. And those are peripheral. So peripheral is on one side only. Oh, you see how much fun I'm having, and it's only the second chapter. I'm going to get better. I'm just going to say that. Integral go crosses entire membrane. And maybe you just have proteins on the inside kind of stabilizing the cell, giving it support. Maybe you have just proteins on the outside as part of a tag, an identifier. All right. So the other thing I have are these little yellow guys here. And these little yellow guys are cholesterol. I love this picture. I could talk about it forever. Cholesterol adds to the stability of the plasma membrane, actually the fluidity of it. Because things flip around and things swap about, that's why it's called the fluid mosaic model. I think that's on the next slide. So my handwriting is horrible, and it really isn't as bad as it looks when I do these things. Okay, so proteins that are able to expand from the outside to the inside will let things pass through either one direction or another direction, so they're kind of also known as a channel protein, but they're integral, transmembrane, they go across. Now, if I have a sugar is like anything with the prefix glyco, so glyco is a sugar. Like glycogen and even glucose. If it's attached to a protein, it's called a glycoprotein. So this guy is a protein with this purple sugar here. So that's what makes it a glycoprotein. If I have a sugar attached to a lipid, like this is a sugar attached to just some tails here, it's a glycolipid. So I have glycoproteins, I have glycolipids. I have cholesterol, I have phospholipids. I have peripheral proteins on one side only, and I have integral proteins that cross from the outside to the inside. Those are all my components found in this plasma membrane. So let's look at the next slide. Okay, so why do I care about this plasma membrane? Because it is an amazing barrier. It is selective. I know that's a later slide, meaning some things pass through. And once we kind of talk about what the deal is, we'll go into specifics about what can pass through and what can't pass through. So therefore, it helps control the flow of what's entering and out of the cell. Certain things you can't control. It is a cell marker, so it's for identifying and then also working with other cells in terms of signaling. I don't have much to say about that. Oh, here is my fluid mosaic model. Some things are free to move, and the main parts of the bilayer are my phospholipids, cholesterol, glycolipids, but then we also saw glycoproteins. And then these other proteins, integral, meaning they go all the way through, and peripheral, meaning they're on one side only. Okay, so let's look at this phospholipid. I'm never going to ask you this structure, but I like this because it tells you that it has its big round head, and then it has these two legs, right? These two tails. Okay, so this is the head, these are the tails. Now, the head is a phosphate. That's why it's phospholipid. And the tails are the lipids. And so you combine phosphate and lipids, and that's why you have a phospholipid. Okay, so the phosphate head is charged, okay? Oh, I didn't tell you that word yet. The head is charged. So because it's charged, it's hydrophilic. And hydrophilic means it likes water. Wow, your body's water, so good thing something likes it. So I have the heads, that like water, and then I have these tails, and these tails are lipids. And what happens when you put a lipid is a fat, all right? I'm going to use that word a lot. What happens when you put fat in water? It doesn't like it. It stays with its own buds. It's like, oh no. And so these tails are hydrophobic. They don't like water. It's water-fearing, but I'm like, I don't feel like it's water-fearing. It just avoids water. Oh, don't like. Yeah, that's supposed to be a K. Water. So when you put them all together, like if I had a whole batch, a whole cup full of phospholipids, and I dump it in a cup, what's going to happen is all the heads, where's my pen? All the heads are going to be toward the water. And the tails, no, I just pushed a button by accident. These tails are going to orient toward themselves, orient into middle, where there's no water. And not only do they orient in the middle where there's no water, but they form this, so it's a nonpolar core, that's the middle. Middle doesn't like water. Doesn't like water, doesn't like anything charged. And things that are charged are called ions. Doesn't like big stuff. All right, so that's a big deal. So nothing big, no ions. We haven't really talked about ions yet. We do talk about them considerably. Nothing can enter into and out of the cell through this bilayer. Unfortunately, does allow some things to go through. I'm not sure if this is a later slide or not, so let's just talk about this. What does it allow through? It allows through oxygen, carbon dioxide. It allows water to go through. And then another thing that's kind of scary is that it allows small lipids. Nope. And small lipids are like steroids. Steroid hormones. So if somebody is on steroids, that can enter into the cell nonstop. Nothing is stopping it. That's a big deal. You can really mess with your body because there's no blocks, there's no guards. It's a barrier. It will not allow things that are charged, that's like ions, and things that are kind of have little charges built into them. And that's why they're called polar, or large stuff. Also won't allow all of that. So phospholipid is a big deal, right? You need lipids. I'm not saying fats are bad because your body needs lipids as part of cholesterol. We saw as part of the cholesterol in the plasma membrane and just gathered throughout. Cholesterol is a small lipid with a steroid base. Then we had the glycolipids on the outside of the cell. So those are on outside only. Extracellular means outside on the surface. So if it's on the outside on a surface, then that is going to be some sort of tag. Oops. And also glycoproteins. So glycoproteins are typically integral. They cross it with a sugar. So carbohydrate is a sugar on the outside. And that crosses it. So integral means that, remember, crosses bilayer. Oh my gosh, this handwriting is just dreadful. Okay, so another big fancy word tying into my plasma membrane is just the glycocalyx. So the glycocalyx are anything, right? Glyco, sugar. Anything that has carbohydrates, either the lipids and the proteins, and just identifies that cell. Because if the cell isn't being identified, this is important. Identifies cell for chemical messengers. It identifies cell because if it's messed up, the white blood cells are going to munch it. It's kind of like the passport permission to be in the body. So, big deal. Glycocalyx, big deal. Okay. Oh, so now we're talking about some membranes. We had the peripheral membranes just on one side, either the outside or the inside, and then the integral membranes that cross it. I think we've kind of gone over that one. So what's the deal with them? Actually, here, I'll let you read that for a second while I take a drink. Ooh. I'm going to talk about I'm just wondering, should I talk about this right now? Yeah, I don't have too much to say about it. All right, let me move my little box here. I'm still in the active window. I learned that lesson. Okay, so proteins. Oh, hello, are you writing? Okay, proteins are a big deal. Look at all these different functions that they do. Okay, and this is the only slide that you have on it because I deleted all the other ones. Proteins, if they're integral, right? So if it can cross from the outside to the inside, they can be channels. They can be channels for ions. An ion is an atom with a charge. It means that it either gained or lost an electron. And, oops, I guess I better write that out. You're like, well, okay, no, I don't need to know that. Well, what you need to know when it comes to ions is that this is like your sodium. Sodium in your body's in the form of an ion with a plus charge. Chloride, sodium's frenemy. He loves them and he hates them. Potassium, magnesium, calcium. I mean, I could go on and on. Oh, I think we saw another one, right? Didn't we see bicarbonate? Yeah, we did. I mean, these are all necessary for life, necessary for nerve impulses, muscle contractions. Oh, it's getting good. It's just you need ions, okay? You need ions. They're critical, so they can't get across because they're charged. And because it has a charge, so it's blocked, so I have to have a way, and a way is these proteins that form pores. I can have, we're going to see this in the next lecture, integral proteins, so it crosses from the inside to the outside, and they're a carrier. So they change shape. They get something, they change shape, they let it in the cell. All right, so these are, we're going to see these quite a bit in the next lecture. Another thing that an integral protein can be is a receptor. So something can come, bind to it, and it just causes a whole lot of chemical reactions inside the cell. This says it alters the cell function. It causes changes inside cell. Inside, oops, there's an E in there somewhere. And another thing is that it can act as an enzyme, causing reactions one way or another. I can have proteins that link things together, anchoring to give the cell a stability and shape, because you have a lot of cells that have a very specific shape, like in bone. And then also, like we've talked about already, is that it's a marker identifying who it is. And, can stay alive, not get eaten by a white blood cell. I want to say the word destroyed, but we'll just say be killed. That big X means be dead by a white blood cell. Okay, so there are all different functions. One, ion channel, letting ions in. Two, carriers. So it's also a channel. It's a carrier channel. Letting in other things that can't just cross, like glucose and sucrose. Amino acids here. Another function of a membrane protein is to be a receptor, to bind to a message. they're going to be very specific. Well, I guess everything's very specific. And it can be an enzyme causing chemical reactions. This proteins can link cells together, link other proteins to the cell, and it can be a cell marker. So 6 functions of membrane proteins. That's a really easy question to ask you on an exam. Okie dokie. So they're fluid because things change, they flip around. They're mobile. So fluidity doesn't mean like it's a liquid. It just means that things move. Okay. Oh, and look, we already talked about this. Awesome. So it's selectively permeable. So it lets in. Permeable means what does it let in? It lets in something. And we actually already just talked about this on another slide. Whoa, no. I need to go back. How do I go back? Is it on this thing? No. Hold on. Oh, it's that easy. My bad. Okay. Things that are small and have no charge, oxygen, carbon dioxide, and steroids. Steroids are small lipids. Okay. They pass through no problem. Otherwise, I need a channel or a transporter to get them through, and that would be like glucose, ions, other amino acids. Oh, we'll write it out, right? What's the rush? Amino acids, all this has to happen. If I'm super big, super large, I have a different way of entering the cell, and that'll be in like the 3rd lecture, not even in the next one. So, here's my question to you. Why can't ions cross? Why can't ions cross? Because they have a charge, and they can't get through. We'll say pass through. Those nonpolar tails, remember? That are of the phospholipid. So we got a little technical with this lecture, and this is kind of a big deal. This is actually a really huge concept. I'm not ready to talk about this yet. Should I? No. Let's call this one and not talk about that yet, because this whole gradient we'll get to. Alright, so this is the end of lecture 1. Hope you enjoyed it. Got a little technical. Take home is what's the deal with cells? And what's the deal with the plasma membrane? What are the components? And why do we need each of those components? What are their functions? So you want to think about that as you reflect. I'm hoping you're taking notes, doing something on this lecture. All right, bye for now.